Nice. Okay, I think we can start now. There's no more people joining. Oh, just one more. Okay. I guess I can start. Hi everyone. Welcome everyone to our earlier career seminar series. Today we welcome our second presenter, Noelia. Noelia is a chemist who leads the artificial intelligence for protein design group at the Center of Genomic Regulation in Barcelona. She holds a PhD in biomedicine from Pompeu Fabra University and has proven protein design projects in Acellera, Pfizer, and leading European research institutes. She developed ProtGPT2 and ZymCTRL and recently led a project that applied direct preference optimizations to protein engineering. In 2024, she won an ERC Starting Grant in develop multimodal agents for protein design. And you can take it from here.
Thank you. Thank you so much. It's a pleasure to be here. And thanks for the kind introduction. Yeah, as Mo said, I'm a group leader at the CRT, at the Center for Genomic Regulation in Barcelona. And I was only briefly today thinking that it's been, let me see whether this works. Now, it was exactly 3 years ago, a little bit more now, that I gave my first talk in this seminar series. And it feels a lot longer than 3 years. A lot has happened in the protein design field, including, as we all know, a Nobel Prize last year. But yeah, I guess you all know this. So I'm particularly excited to show you what we've been doing since then. For some reason, now this works. If you're here, probably you know that proteins are the most amazing machines. This is probably a needless introduction, but proteins can do whatever come to mind. They allow us to breathe, speak, digest, and think. And they have also incredibly cool biomedical and environmental applications. For example, they can capture carbon, CO2. They allow us to sense smell. And enzyme engineering has allowed us to also have efficient plastic degradation from enzymes. But you are also possibly well aware that this has been remarkably challenging. So to design proteins that are as active as those that I was showing, that come from natural sources, but that are completely human-made. And I like a lot this work that I'm showing. It very recently came out from the Chris Wellswood group, the Protein Design Archive, because it really highlights the evolution of the field in the last years and how complicated it has been to design proteins. So we see that in the entire protein data bank, we have over 200,000 PDBs, and only a fraction of them are actual designs. So designs that are made from researchers. So it's a tiny proportion of the entire PDB. And then further, those designs are mostly structured. So they are, proteins floating around in vitro, but they perform no function that could be useful for us. There is, however, hope. And we discussed some of this in this opinion piece very recently. But in this paper, Chris and colleagues, they really summarize it very well. So if we look at the pace of development in protein engineering in the last years, we see an improvement of the pace at what we can design we can produce the new enzymes. And this has been particularly changed in the last 3 to 4 years with the advancements in artificial intelligence. So I like to summarize the advances that we are seeing in protein engineering and design in 4 categories. We used to have very good physics-based methods, like for example Rosetta, which model a protein with pairwise interactions, and then we can imagine that we have this huge energy function that it used to be by itself also very approximate of the real forces that occur in nature, but it's already very complex, so complex that it's an NP-hard problem to search for the most minimum position in the landscape. So people were using approximate methods like Metropolis Monte Carlo to go searching, going downhill in this landscape to try to find for the most optimal sequence that falls into a specific backbone. And since then, since like 2000, 2021 more or less is the new era of AI models. We now have models like Protein and PNN that are very good at producing sequences for a specific backbone, but also we have a bunch of other different methods that are not only allowing us to generate for a specific backbone, but also can produce entirely new backbones, like here a contact map. So we can diffuse entire backbones. We can also generate sequences directly and condition them to have a specific function. And we can also condition on specific properties, like in here, we can scaffold a protein around a specific fragment. So we reviewed recently how many of these methods were developed recently, and only in the last, so in 2023, we already counted over 100 methods that have been developed in the last 3 years alone from that time. So the explosion of methods has been really outstanding. And they seem to also be working very well. And it's no surprise because proteins are very particularly amenable for artificial intelligence models. It's very obvious, and probably we have all observed that proteins can be represented as in language because they have an alphabet, which is the amino acids. And these amino acids form secondary structures that combine themselves to form complex domains that carry a function. This is the same as words combining themselves to form sentences that carry a meaning. And these sentences form paragraphs and longer texts that have meaning over longer text. And it's the same as domains combining themselves to have quaternary structures. And also another very obvious application that we've seen is researchers taking models like diffusion models that originally were trained for image generation or image processing, and we see them being directly applied with very good success rates for proteins as well. And it's because proteins, they are text, but also they fold into specific 3D shapes. So they have X, Y, and Z coordinates for each specific atom, which means that we can represent them as 3D matrices in the same way that we have a picture which has 3 channels like red, green, and blue with specific pixels for values for those 3 channels. So it's exactly the same. Whatever the method has been working for the diffusion of images is going to work for molecules like proteins. And we're seeing recently more and more work in the area of trying to either predict or generate time series. So proteins, we tend to forget that they are dynamic, but their dynamic nature makes them functional most times. So it's very important that we also integrate this modality into our training process. And then this is also seeing all the progress we are seeing in deepfakes lately. This promises to be a very prolific area of research in the months or years to come. We specifically focus on the third category, which is protein language models, because of the obvious similarities that proteins and languages, human languages have. And this has shown to work very well. It has some limitations, but I also see that the field is coming to overcome them slowly in the last months. And I'm gonna present you a little bit of what we've done in this area. Probably you all know this, but in case someone needs a review, I will mention that all the area of research that we are seeing in protein language models, so in PLMs, they come from the original transformer model that was developed in 2017. It was a response or a work performed by Google, and they wanted to improve Google Translate, which at the time wasn't as good as, nearly as good as it is today. And the model had, the original transformer architecture had 2 modules, an encoder that is able to produce input text. So it processes that, and through cross-attention passes that information to a decoder that produces an answer, like could be a translation. But in summary, it can be amenable to any sequence-to-sequence task like summarization, translation, or question answering. And then a few researchers, and one of the first ones was OpenAI. They decided to shed entirely the encoder. They took the decoder and they observed that by training the model on huge amount of text, the model was able to speak fluently language. And this was the first time that I also saw the GPT and GPT-2 producing human language so proficiently without any prompts. The way that this works more specifically is that the language models are explicitly trained to approximate the probability distribution over a sequence of tokens, as we can see here. So this is the probability of a given sequence, and then we can decouple that into the probability of the specific tokens. The probability of a given token, like this, we compute it in decoder-only models. So when they are trained with this autoregressive objective, it only depends on the previous tokens. So the model, the attention is only focusing on the tokens previous to the current one at every training step. And this done over thousands and millions of sequences allows the model to learn a probability distribution over sequences. And at sampling time, we have a model that is able to sample from that probability distribution. And it's exactly the same with proteins. So we can take a protein sequence, and then the tokens can often be the single amino acids, and we can then learn the probability distribution of amino acids. After training, the models produce sequences that we observe are in silico very similar to what we see in nature, and also experimentally tend to behave very well as well. We've trained several models and given an overview of the first two and how we are now using them in more real-life protein engineering campaigns. The first model was called ProtGPT2 and was more of a proof of concept to see whether by training PLM directly on the entire universe dataset, it will work and produce sequences that are meaningful and, like protein-like, like nature-like, that they could have the potential to one day be a function. And then we train a second model called ZymCTRL, that stands from enzyme control, that instead of producing sequences in remote regions of the protein space, it can be conditioned to specific function tags, like, for example, carbonic anhydrase. So the model specifically produces sequences for a given condition, in this case, enzymatic commission number. So it specifically generates in a specific region of the protein space. We've trained a third model, but I'm not going to focus on this today. The third model can add a further layer of control. Instead of giving it as a functional tag, it can take an entire chemical reaction. So this allows us to change the substrate or perform modifications in non-chemical reactions, but also will potentially allow us to insert any reaction that comes to mind, including new to nature. Just to give an overview of how the models are trained, I'm going to show how ProtGPT2 was trained. So what I did is to take Generative 50 at a time, which had almost 50 million sequences. And then I didn't tokenize at the time at the character level, but I used BPA, so byte pair encoding. And this gave me a few, I chose 52,000 tokens. And this gave me tokens of around 3 to 4 amino acids. So they were k-mers of that size. Which had a lot of advantages at training time because my window, the context window was a lot short. So I could put a lot of sequences in a single context window with 1024. After training over 4 days in a cluster having 128 800s, this led me to ProtGPT2 for obvious reason. I had taken the original GPT-2 architecture and trained it on the entire Generative. And then after this, I wanted to see whether the sequences were any good. Usually with language, we will just look at whether the text reads well, but we speak English. But here we don't know whether the sequences are good. So we had to come up with experimental and in silico characterizations. What I wanted to show here, which was most remarkable at the time, was that well, at the time I was disappointed that the model, if this is a representation of the entire protein space, the model, I was hoping that it would generate a lot of new islands. So each of these dots in here are a component in a network that represents the sequence space, and every component corresponds to sequences that are linked to each other whenever they are above a certain threshold in sequence identity. And I was hoping to see a lot of new islands, like sequences that will be completely novel and remote in the sequence space. And I didn't see many except for a few instances, like here. This has only like 9% sequence identity. But I saw that there were around areas, so like clusters that we already know. However, what was very exciting to see is that for the first time, we were seeing proteins that have embodiments and loops. And this was very important to for us as a reinforcement to see, oh, they have the potential to have function and like to be flexible and accommodate ligands and other binding partners and potentially even be enzymes, because most of the protein design campaigns to that day, they had been producing proteins that had very short loops and they were very idealized, and that made them very rigid and very likely to harbor any function. So we were happy. And then we proceeded to train the second model to also go for the lab and check experimentally whether it will work. The second model in nature is very similar. It's a decoder-only model as well, but it's conditioned on enzymatic commission numbers. And what we did here in collaboration with Basecamp Research is to take all the enzymes in the UniProt data set that contain an EC label, so that they have been annotated as enzymatic commission, so that they are enzymes, but also that they have a specific annotation for the enzymatic reaction that they perform. And then I prepended them to every sequence and tokenized them at the EC label as well, at that level, so that the model could infer that whenever there is a 2, these are oxidases, and whenever there is a 7, these 2 sequences have up to this level of similarity in the chemical reaction that they are able to catalyze. I did this because we observed that the data set that we have in public databases is very unbalanced. So we have lots of points for sequences with, for these labels with as many numbers as in here, over 1 million sequences for this is label. But for other is labels, we know very little, and we have maybe only 1 member. And this causes a problem because it's unlikely that the model will generate properly from those cases in here. By doing this, we can transfer learning from those with the label 2 that are more populated than others. We tested this in 4 scenarios. So I'm going to show you 2 works that we did, 2 testing cases that we did in the lab ourselves, 2 cases that other people did as well using the model. The first one was carbonic anhydrases. We wanted here to see, well, this is one of my favorite enzymes on Earth. They are able to capture CO2 very quickly. So every carbonic anhydrase is capable of capturing CO2 more than 1 million times per second. And this is pretty amazing because as we know, we have plenty of CO2 to capture. So we generated 20 sequences from which 7 showed activity. We were very stringent in the sequence identity threshold, and isomerase confirmed that we had 7 of them that have shown activity. Then Ramiro, who's our lab manager in the lab, did the structural characterization for these 7, including CD spectra and segments, and showed that they were monomeric. The second example that I want to show, it wasn't not performed by us, but I particularly like it because they also were able to produce enzymatic enzymes from ProtGPT2, which had not been conditioned or trained on enzymes. So whatever is on the Generative data set. And what the Hooker team did is they took ProtGPT2 enzyme control, they generated triose phosphate isomerase enzymes, and they cluster, so they filter them to be maximum 35% sequence identity to any natural sequence. So this is a very stringent cutoff. And they tested them very elegantly in complementation assays, which works like this. So we have an E. coli version that lacks the wild type triose phosphate isomerase. So whenever there is not an active enzyme of our designs that is plugged in there that is going to be active, the cell will just die. So the designs need to be active for the enzyme for the bacteria to survive. And this is what you see here. Out of the ones I tested, they saw a few that were active. So you see, I don't think you see my mouse, but so if you read in reverse, Spoken T9, that was one of the very best ones. And then they took that one and further purify and characterize it structurally, and they observed that it was almost as active as the E. coli wild type at team barrel, the team wild type enzyme. So this was pretty outstanding to see because the sequence identity for this particular enzyme is below 35%. In here, we have done a few, up to here, several assessments for these enzymes, so including carbonic anhydrases, lactate dehydrogenases, and the triose phosphate isomerases. But we were always doing them in like batches of a maximum of 10. So we were always designing sequences of like batches of 10 and testing them in the lab. In here, they did a few. We also did a maximum of 20. And this didn't ever give us like a statistical overview of how well the method works. But then the protein engineering tournament came. This is an initiative from Align to Bio. And what they allow us is to different teams send submissions for testing. In that year was the specific case of alpha amylases, but I think this year there's going to be a new round for a different enzyme type. And we submitted 2,000 alpha amylases, 2,000 designs that they tested in the lab for us, purify, express, and gave us the activity values. And this was very interesting to see because initially we were very happy that we had a few or several enzymes showing values over the wild type levels. But then when we look at the entire picture, we observed that the actual training set that we had trained the model on also had levels that are as active as the wild type or more. So with this, we were thinking, well, it seems that the model is simply replicating the activity levels that we have in the training set, but usually we want to be over here. So if this is the activity axis, we want to generate alpha amylases that are more active than anything that is in the training set. We want, to engineer enzymes. And this was not only for the activity of the enzymes. So we observed this property happening across several other features like lengths. So we see in blue is whatever the model, so the properties for the model for the generation, for the generated sequences. And in gray, we see the properties for the training set. And then we see that always the blue distribution mimics perfectly the gray distribution. So we see the model, which is good, learns to capture the properties in the training set, but it won't be able to surpass that beyond what's being seen in the training set. This is good for sampling, but not for engineering. In fact, this has been also reported in other domains. So this was a very nice read. I recommend everyone here who's interested in PLMs or generative models to read the Nature paper that you see up there. And this is that models collapse whenever they are trained recursively on synthetic data, but also the first epoch they are able to produce the same. So they collapse to the most common distributional modes. So if you look at this, for example, at the picture of the dogs, you will see. Imagine the training set has a very wide distribution of different type of breeds. So you will have the golden retriever, Pembroke, and different sort of breeds for dogs. Then whenever we generate, imagine there are pictures, we generate from the model in the first iteration. Already the model will sample most probably from those that it has seen more often. So we will get mostly golden retrievers. But if we keep fine-tuning the model over time with this with its own generations, over time the model collapses and produces sequences that are just, or dogs that are just the same, and up to a point that just collapses and produces things that are gibberish. This is what we observe as well. So in the first iteration, the model mostly reproduces the most normal modes in the training set. But also Maria went ahead and checked whether we will see the same by reproducing. So by taking the best sequences, imagine we take them a PLM, we generate sequences, we take the best ones from every batch, and then we fine-tune the model with those with the hope that it's gonna be getting better over time. But this is not what happens. If this is the values that we want to optimize, we see very little improvement over time. Imagine we want to optimize something like PLDPT, we see no improvement at the expenses of the sequence length, in this case, getting shorter and shorter. So we see that this happens for PLMs as well. And then the question is, how do we optimize from PLMs or for generative models in general? Let's remember that whenever we train a model like a protein language model, what we have is in the end the probability of a given sequence for given a certain set of conditions, like it will be for us with the previous models like the easy labels. We learn this probability very well because we have millions of sequences. So the model learns unsupervisedly from those millions of sequences. And in a sense, it learns some notion of fitness because it sees a lot of variation across the species, but it's not aligned with the engineering fitness that we want. We have no data for how, for example, those millions of sequences will be behaving under certain scenarios like the activity of the alpha amylases. So it's fitness in the sense of survival, but not for the engineering purposes that we need. The question is, how do we maximize for the fitness that we want? we can define a fitness function that is a scalar that for every, we associate a given value for every set of sequences and their given conditions. Like for example, imagine the alpha helices for a given sequence, then we have the given easy label, and then we associate to it a given activity value. And now the question is, how do we maximize not the probability, but the fitness so that the model generates a sequence that maximizes this other function? And we are seeing a lot of work in the last only 5 months in this area. One of the I'm thinking of this in 3 different categories. One of the earliest things that people were doing was to optimize in iterative fashion. So this, for example, was a very pioneering work by the charts lab, but led by Biswas and others, in which they took, at the time, UniRep, but it will be the same these days if we take ESM or ESM2 or any other encoder only model, and then we train a regressor like a top layer that on a set of activity data, and then we can sample from the model, assign a given activity, and then do mutations on that sequence, fed that to the model again to get what is the new activity, and then we can accept it whenever it's better than the previous step. So we go doing like little steps, mutations, and it's sort of a hill climbing algorithm. But by doing mini steps, we will be able to go from the start to the end, as we see here. And this worked for them very well with very few data points. We also see success in steering. This is a very recent preprint that I enjoy reading, in which, in this case, what we will have is, for example, a model that is frozen, like a diffusion model, and then there is a steering process in which, like, a value function that has been trained on activity data, for example, it's inserted into the process of the diffusion, like, for example, classifier guidance, and this allows the model to guide. So this allows to guide the model towards a certain generation. There is also what we are going to see today here, the process of reinforcement learning. There is, this is what I'm going to show you now. There is a big difference between all these models. So in these 2 cases, the model stays frozen. So the PLM, for example, will not be modified. The weights are the same. In reinforcement learning, what we do is we iteratively improve the model with feedback. So it's different paradigms, but the objectives are the same. We try to get the maximum possible fitness out of a given function that we want to engineer. What we do with PLMs then, so the paradigm using reinforcement learning with PLMs is more or less as follows. When we have a PLM, we can sample very large landscapes. So with this, we have a very broad coverage of the entire brain space. This is nice because previously we were using engineering tools that would allow us to explore the local space, but maybe not regions as far away as we've seen with 35 and 40% sequence identity only. But we want to reach to the top. So what we can do is to generate those sequences, test them in the lab, and then reinforce the model with the feedback. So whenever we have good sequences, we tell the model, we punish the bad sequences, we reward the good ones. And by doing so, the model is able to iteratively improve, and we should be able to get here to the top. This is rooted on agentic workflows. An agent is by definition a system that senses its environment, which in our case is the experimental setup, so like the testing itself. It takes action, which will be, oh, I'm going to improve in this direction because the sequences that I generated in the past were not as good as they should. And then it improves over time with this feedback. So the model should be getting better and better over time for this task. More in practical terms, what we do is we pre-train a model. So this was done. The first step is we pre-train a decoder-only model on millions of sequences in a self-supervised fashion. So like we've seen with ProtGPT2 or ZymCTRL. Then we can do supervised fine-tuning on the specific set of sequences that we would like to optimize. For example, it will be alpha helices. And then with that, we are able to have the baseline. Like the model is going to explore sequences that are active at the level of natural ones and are very diverse among themselves. And then we can apply the reinforcement learning paradigm in which, the model generates some sequences, we test them in a certain environment, and we apply rounds over time where the model generates better and better sequences. More mathematically, and here I need to say that all this work was led entirely by Filippo Shitoko, who I think, by the way, is in the audience. And he's now starting a PhD in the group with us. So Filippo, what Filippo thought is about applying direct preference optimization that had been applied very recently to what we see in online language models. So as opposed to PPO, in DPO, in direct preference optimization, what we have is that the model originally will be aligned to first imagine that we have a sequence that is behaving good in the lab and a sequence that is not. Then the model should learn that the log likelihoods for the good sequence should be better than the sequence that is not. So the model aligns itself with the results. It should maximize the distance between good and bad pairs. And we do so by preventing as well that the model deviates too much from the original training distribution, from the original reference model, because it was already a pretty good model. Like, we don't want to entirely catastrophically forget everything that the model had learned. By applying so, Filippo was able to optimize many in silico properties, but also experimental ones. For the sake of brevity, we can very briefly skip this, but you know that, so the PLMs that we work with, as I mentioned before, they take an easy label, we generate the sequences, and then the environment can be anything. It can be an oracle that either tests the topology or the activity in the lab or the stability, or it could be AlphaFold's PLDT. Anything can be an oracle, and you can also combine many of them and then feed that back to the model depending on the performance of each of the sequences. The first property that Philippe optimized was topology. So imagine that we have the original sign control or a PLM that for a given label like this one, or in general, produces more sequences that fall into the beta-carbonic anhydrase, like in here versus another one. So in short, whenever we input a model like a sign control with the label of the carbonic anhydrases, it has a tendency to produce more beta-carbonic anhydrases than alpha-carbonic anhydrases, simply because there are more beta than alpha in the training set. And the proportions are more or less like 60 to 20%. So one quick way to check whether DPU will work is like, can we shift the generation from beta to alpha, even though it's preferred to generate beta because it's simply more favored by the model? And indeed, Filippo managed to go from the proportion of beta to alpha and revert it in less than 10 epochs. So we see here the proportion of sequences as computed by the TM score. So we get the TM score for the alpha sequences, and over 5 iterations or so, the model will reach the topology that we want. So it will be going from beta to alpha. This is not happening at the expense of sequence diversity. So we see that even though the model is deviating from beta to alpha to this new portion of the protein space, it still generates sequences that are very diverse. So for every iteration, Filippo is generating 200 sequences, and then these are how many clusters are there are 90%. So this is still, most of the sequences are still diverse at the 90% level. Another thing that we can optimize is how well the sequences are supposed to do in the function that we want them to be performing. So if we generate, for example, sequences that perform this catalytic function, and then we take an external oracle like clean, which is a model that is able to annotate easy labels for sequences, we will observe that sometimes for those labels for which we have not many data points in the training set, the model doesn't generate sequences that clean will assign with the correct label. This is an issue because then we need to generate lots of them, and sometimes it's a problem whenever the model doesn't even remotely generate the label that we want. But by applying DPU using clean as an oracle, we're able to revert these proportions. So we are able to go from like a percentage of correct these labels around 10% to 20, and then push that to over 50% and 80% in a few iterations. So the model is able to recover the predicted functionality even farther. Something that we observe is that if this was the level of correct easy labels at the baseline of the iteration 0, this is at iteration 30 for this easy label that we were optimizing. But we observed that other labels were improving as well, even though that only this easy label had been rewarded. So somehow the internal distribution of the model changed sufficiently to also allow for improvement of labels that were neighboring to the original one. This also happened for PLDTT. So this was the original easy label that we have been optimizing for. Also, other labels optimize the PLDTT values over the iteration process. Then we put this into the lab, into the context of real engineering. What we did is we were very lucky to just be in time for the adaptive bio tournament. And then Filippo submitted 6 sequences, from which 3 were nanomolar binders, 2 of them being better than the baseline, which is the wild type EGF. So what we see here is the lower the number, the better, because we want to optimize the dissociation constant. We recently participated in a preprint. So here you will have all the analysis for the results for all the submissions that the different teams submitted to try to optimize binders for EGFR. But then this gave us several data points to optimize binding affinity. We want to reach the lowest possible numbers, but this gave us several data points, including those from others in the tournament, to improve the model over time. So what Filippo did is to generate a second round. So round 1 was the baseline when the model had not been optimized or aligned with experimental data. The second round is after optimization. So once the model has been rewarded with experimental data from our results and those of other participants, and then we reached overall a much better mean results for the dissociation constant, including one binder that is very low nanomolar, in 27 nanomolar. This here to the right, we can see the difference between the first round and the second. So we had several binders in the second round that were surpassing the wild type, but also the binders in the first round, as we would expect. And also we can see that with the model or the generation process keeps exploring sequences that are diverging more and more from the original point up to around 2 or 3. So we start very close to the wild type, but then as the model goes exploring through the generations, we generate sequences that are farther away. So this gives us hope that we're exploring larger landscapes. And then I want to also highlight the work that we are doing with others. I'm very excited that we recently got an open trust. I'm sure for many of you this is very common, but for me it's one of the coolest developments in the recent years. So we can have these robotic platforms that are completely programmable in open source. And by doing this, we have entirely here our environment. So this is literally an environment where we can test our models and get the rewards. So what our collaborators did, so Marwell and Maria and Edu, including as well with Filippo, is they take several agents, including time control, ProtGPT2, but also other models, and then they can generate thousands of sequences that get tested into in an oligo pool assay. In this case, we were trying to optimize DNA binding, and this high-throughput screening produces lots of data points that can be fed back again to the model. And by doing so, over runs, the model should be able to produce sequences that are binding with higher affinity to DNA. One thing that I would like to mention before giving time for questions is that we see a little bit more of explainability work in the recent years or months, but not so much in the field of protein design. So what Andy did, who's a PhD student in our lab, Andy reviewed over 150 recent works in the field of explainability and wrote a piece where we can see how these different technologies, how the different methods could be applied to protein design as well, so to the specific case of PLMs that are used in the generative case. But going back to the previous work, we can see that some of these methods are already directly applicable to what we were doing with PLMs. In particular, so Filippo took, for example, one of the best binders that we had in the second round, or the best binder. And in here, we can decouple the reward that is assigned to the entire sequence to the specific amino acids following this very incredible good work from Rafael, and others. The way that this works more in detail is by imagine that the model has seen this entire sentence in which is a Reddit post in which someone says, oh, the expectation during the interview for a certain salary is this number. And then something happens. And then the second sentence says, oh, the expectation is for this review a different number. And then the model, when we look at the decomposition of the different rewards for each of the tokens, it clearly assigns a different value of reward that you can see the different colors to the value of 38K versus the value of 250K. Now you can see my mouse, I hope here. So this is the biggest difference. And we can do the same over the process of reinforcement learning, but for our tokens, which are the amino acids. So we can see over the process of learning how the model decouples the specific reward for each of the amino acids over time, and even produce mutants. So what we can do is, if this is the wild type, we can mutate it to one of the catalytic residues to another amino acid, and then we will see that the model is capable of assigning certain credit, or it has lower reward in this position because it shouldn't be there. Like that's not something we expect from a functional protein, and this happens at the without changing the entire reward distribution for the rest of tokens. So it's very good for credit assignment. Like we can pinpoint amino acids that should be critical for a given oracle, like in this case, activity. And going in this line of explainability, something that we explored recently, that Gerard entirely led, who's a master student in our lab, is the use of Spartan to encoders for protein design. Spartan to encoders are, although many of you, I'm sure that you've seen many of the very recent works applying them for encoder-only models. They consist of an encoder-decoder architecture that they learn to produce this intermediate activation of higher dimensionality. So the higher dimensionality is made on purpose. It's incentivized to be sparse through the training process. And this allows going from very polysemantic features to atomic features that should be a lot more monosemantic. And this will allow us to extract more humanly interpretable features that theoretically map into properties in the protein, like structure or perhaps even activity. So what Gerard and Filippo did, they took up a DMS for which we had data in the alpha amylases. So for every sequence we have single mutants, and then we have the associated activity values. Took a fine-tuned model on this data and trained the Spartan to encoders on that, with the hope of extracting features that will differentiate between active and inactive sequences, so that we can extract what features in this space are making a sequence to be active. And then these interventions at inference time. So there is clamping and ablation. Clamping involves setting the activation of features that are identified as positively correlated with activity to their maximum in the training set, the maximum that was observed. And then ablation is doing the opposite. So basically setting to 0 during inference time those features that are negatively correlated with activity. And then by doing these interventions, that we can tune then mildly the activity. We don't expect from a single feature to rule the entire activity prediction, but we see some modification only with doing clamping and ablation of single feature. So there is hope that in the future, by doing combination or genetic algorithms perform on those, that we will be able to have better control over the guidance of the model using sparse autoencoders. And going from the overall, what we dream for the futures to come, for the months to come, is to train agentic models that learn from many different modalities, including protein dynamics, structures, chemical entities, and annotations, and that we can test them in robotic platforms directly in the lab. And with that, the model is able to improve over time for the biotechnologically relevant enzymes that we would like to design. And yes, I couldn't present any of this without my amazing team. Here are all the current PhD postdocs and technicians working in the lab. Also, I have infinite gratitude to the funding agencies that are allowing us to perform all the research that I showed here, and also thanks to the many collaborators that we have. And also, we are here literally working in front of the beach, so reach out if you would like to hear about open positions anytime, and I'm happy to take any questions.
Thank you for the great presentation. I guess we have only one question, and we have two questions in the chat. As first question is, even though the sequence identity looks not verified, but they're like still relatively high, And I think the fact that the structures seem pretty similar as in the primal images, are these designed like synthetic homologs of the natural proteins?
I agree. We will still consider that given that the structure and the pocket that are preserved, these are not de novo sequences. So this was one of my, let me share my screen again. What I was hoping to see, do you see my, yeah, or, okay. What I was hoping to see when I trained the first model was that the model will explore sequences that are completely far away from everything in nature. But as we've seen later in the talk as well, the model reproduces everything that is in the training set. So it would produce sequences as well that are mimicking the training set. So if the training set is clustered at the 50%, the sequences are also around the 50% with a certain radio. So, I absolutely agree. For de novo enzyme engineering, I think we need different types of strategies that we are looking into. But I see most promise on whenever we introduce geometric inductive biases like diffusion models, as we are seeing from some of the recent works in the Baker lab and others.
Thank you. Leo have a question in the chat box in slide page 38, how to reinterpret the result plot.
Let me see slide 38. There we go. Yeah, so here, whenever we do clamping, we will expect the model to activate that feature. So we will expect the model to produce higher predicted activity values. We see that over the baseline, which is, so if you look into the model to their distribution in gray, that's what the model produces when the base model produces as the range of activity values. For the features where we clamp, we will expect it to improve slightly, move towards higher activity values. It's not a really huge improvement, although we see that the number of sequences that we need to sample from each of the models, so like from the base models versus that is activated for feature 5640, it will be fewer sequences for the case of that feature to reach, for example, an activity of 6. But I see, and this is a work in progress that I only wanted to show for you to see what ways in which sparse autoencoders could be applicable for protein engineering as well. I see more, perhaps in the months to come, more progress into engineering for more activity, at least in silico, by combining either features or other optimization algorithms, or including things that we are looking at, like genetic algorithms.
Okay, thank you. Another question in the chat box. It's like how many examples are used in each iteration of this active learning process?
In the active learning process, it can vary. And Filippo and Lasse, another PhD student in the lab, also take up what would be the trade-off of like how many sequences do you need to generate to achieve a certain activity value over time versus the trade-off of how many sequences versus how many iterations. For the specific in silico benchmarks, we need 200. For whenever we do experimental assays, we are testing 20 to 25 at a time, except for that I showed with the open chance, in which we can generate many more because it's an oligopool assay, so we can test up to thousands.
Okay, thank you. Another question is for the conditional model, one of the main obstacles is having a reliable regressor for the function of interest. Was this part of design or implementations in the enzyme control, or is this something already available?
Here, so for the regressor, I guess, yes, I agree, or also we call them oracles. It is very important to have reliable oracles, and it's the most critical part. For design control training, it's also unsupervised, so the easy labels are associated to the proteins already in the databases, and they say nothing about how active the enzyme is, only whether it's a binary classifier, whether it performs the activity or not. So in that sense, that was relatively easier to train. We've seen other models from other groups like Pascal Notin and others that were directly infusing the activity data into the training of the model. the conditional training of the model so that you could steer the model directly with, oh, I want to generate higher or lower activity values. And I imagine this is a very successful approach for steering the models. But yeah, for this this is a 2022 model. So for this case, infusing the labels directly as they did in the databases work. Also, other PLMs are doing it like this. So using Go terms, for example.
Very cool. Another question is regarding the SAE, the spaced order encoder. Could you explain on how genetic algorithms could be used in this context, and how could this approach be made interpretable?
Answering the second one first, I argue that sparse autoencoders, although they help us to maybe extract features that correlate with properties that we want to see, I don't personally see them as interpretable as one would hope, because if you look at many of the papers coming out these days with the sparse autoencoders, whenever we have 7,000 features and then we map them into the structures, it's very complicated to see what they really are mapping to. So we may see, yes, some of the features seem to correlate periodically with, oh, they seem to be in alpha helices every 4 residues, so they periodically correlate with that. Some others are incredibly abstract. So for us, I would say it's harder to interpret. Genetic algorithm is honestly on the record of PhD student in our lab who had the idea and who's having a look into this and how we could clamp or ablate by combining several of these features at once, and then combining imagine sequences that all have these features and they are active, and then we produce iterations over time. But it's not done yet, so it's any other optimization process could also be perhaps amenable. There is a lot of room for improvement, I believe, and I want to state in using XAI for protein design. There is a lot of work that is happening in XAI that we haven't explored in protein design.
Thank you.
Thanks to you.
We have covered all the questions in the chat. If anyone have like new question we haven't covered yet, you can just mute yourself.
What kind of synthetic data will be used? Xiaotong, I don't understand well. Well, synthetic data is whenever we generate from the model, that's synthetic by definition. So we generate a first iteration that is synthetic, then we test that in the lab, that is fed back to the model. So the model improves over time with no natural data, it's all synthetic. And yeah, so it's also very good in terms of that we don't need for all the in silico optimization to add extra annotation or data from databases or do curation, because the model learns from its own process of generating more and being tested over time. If that was a question, which I don't know. Okay, a few more questions. Renhao, I'm not sure if that's covered. Just wondering if you tested your model on designing protein binders for membrane proteins. Yes, incidentally, although it was tested on the soluble version of EGFR, EGFR is the epidermal growth factor receptor, and that's membrane bound. And it was part of the adaptive bio testing tournament, so the binding tournament, or binding competition, sorry. And then the binders that I showed that Filippo had designed, they were binding to the membrane proteins, yes. There is no limit to what we could design as long as we have the good oracles for that.
Oh, we have more question on the oracles for steering models. And the challenge is getting these ranked dataset, which typically have to come in pairs of triplet, which is challenging for many of the existing large functional annotated protein dataset. Have you thought at all about how we might make use of these larger non-ranked datasets for steering PLMs to certain functional regions?
Excellent question. I didn't explain that very carefully. So even though the original DPO implementation has, it uses the pair version in which we need to couple into, as you mentioned, good to bad sequences in a sense for a given property. or we use the weighted version in which for every sequence we can couple directly the value that we are rewarding with. So every sequence gets coupled to its reward value itself, which could be the activity along with other parameters. This can be, so it can be several oracles that get combined into by, for example, doing a linear combination of the terms, normalizing and then passing that entirely as the full reward. So non-ranked datasets are also perfectly, that's almost in all cases what we do, are perfectly amenable for reinforcement learning campaigns as well.
Here's another question about the explainable part. We have seen a plot for the reward distribution. Was this compared against the general PLM's block probability to check how they compare to each other?
Yes, yes, the baseline is included. But again, the gains were not massive by simply intervening on a single feature. But yes, it was compare versus, so there were a few of the features that were significantly affecting the distribution of activities for the predicted activities. But yeah, the base model is included as well.
Okay, cool. I guess we have covered all the questions. Thank you very much for giving us this great talk, and we're really looking forward to your next big project.
Thanks to you. Thanks for having me. Yeah, happy to